me this opportunity to just talk a little bit about my project. So I'm a graduate student at the vet school at UB, and my supervisor is Professor Ora. I know some of you all might know him. I think his wife comes to the meetings as well. So I just want to talk to you all about, I haven't yet done my testing and analysis of data from samples that were collected on the Tobago Island. So I'm just going to give you an overview of some of the things that we might find, talk a little bit about viruses and what my project is about. So this is my project summary. So it basically is it's a surveillance study for among the avian populations, that's both poultry and wildlife, to look for potentially high impacting respiratory viruses. So the background, as most of you may know, or some of you may know more than I know. I know I don't know much. So there's little published data on current circulating viruses among the populations, even in the poultry industry, which is so large here in Trinidad. Very little is known about the reservoirs as well. We have so many isolated populations, like in Little Tobago, some down in Orokun area, and we don't know what we don't know what's there. And then of course there's a lot of bird migration taking place, as you already know. So things might be coming in, and we want to know what's there, so just so that we would be more prepared if we have to take any action. So I'll just go into a little bit about what viruses are. Most of you might already know. So viruses are obligate intracellular parasites. They must have a host. They are not living organisms, and they require cell components machinery. So that's why they basically hijack cells. And all living cells are susceptible to infection. So birds, humans, any animal life, plants, and even single cell organisms like algae. So the characteristics basically of a virus, there's genetic information in there. There's DNA or there's RNA, and then there's different types of DNA and RNA as well. They're very small. Of course, they cannot be seen with the natural eye, and they are metabolically inert, and they rely on having a host cell. So this is the basic virus structure, and what like the glycoprotein, that surface protein, that's what is sort of the feature that allows it to attach to certain cell types, and different receptors. So that's why the different viruses infect different hosts and different types of cells as well. Okay, go ahead. So this is just a picture of some. I guess you all can tell me what they are. Yes, they laughed at me actually, because my friend went down in the mud when I was going to take a picture, and they were laughed. It was hilarious. So you all know these pictures, and Darshan would have gone through to mention some of the birds that we have on Little Tobago Island that were sampled. As a matter of fact, the only ones we handled and sampled were the red-billed tropic birds and the Audubon shearwaters, because of their because they are on the trails, and we're able to get them because of their breeding in those areas. The brown boobies and the frigate birds and so on, on Little Tobago Island, the frigate birds aren't on Little Tobago, but the brown boobies, they are in inaccessible areas. So we kind of, as Darshan said, we'll have to learn some mountain climbing first before we venture there. Yeah. So why viruses, right? Viruses, as we said earlier, they infect, they can infect any species of life. So no one is excluded, and many viruses have a zoonotic potential, And you all have seen it for yourself these days. Most emergent infections, they originate from an animal or insect vector host, right? 70%. Seabirds are usually in more isolated environments where there's little human impact. So in terms of, let's say, poultry, those birds are vaccinated. Most of them are vaccinated against Newcastle disease virus, and so birds that even interact, because most of the vaccines they use are live attenuated vaccines. So if other birds come into that environment, they can actually acquire immunity against certain viruses. And of course, seabirds travel and migrate, as we saw from some of Darshan pictures. Although we don't know if it's correct. Some of them went south, but a lot of them went very high up north. And so there's a vulnerability in that in that seabird population as well as in waterfowl. Most of the work that I'm going to do, it will be more so in waterfowl rather than seabirds because of the difficulty in capturing seabirds. So you already know this poster, and apparently I think it's the blue and teal that comes down here regularly. The ladies at Pointy Wildfowl Trust, they've mentioned a couple of times that they do see them around on the trust, and I saw that someone had posted on the TNT Bird Alert that they found a blue and teal at the side of the road at one point, and they took it to a center, but it later died. But these are the kinds of cases, opportunities that I would, try to get samples from, because of, just because they're there already, you could handle them, not having to go out and use nets to capture them. So the focus, my project is two parts, so it's poultry and it's wildlife, and the wild bird aspect of it, we would want to be sampling from the majority of the wetland areas throughout Trinidad and Tobago. It's proving very difficult, but because my project is so large, I could kind of downplay some areas of it. So the main viruses I'll be looking at, some of which there are poultry specific. So Avian Influenza and Newcastle disease, those infect wild birds. But then there's also infectious bronchitis, Avian Metapneumovirus, infectious laryngotracheitis, infectious bursal disease, inclusion body hepatitis. So these are some of the, especially the ones lower down, these are some of the viruses that they vaccinate against, or they don't vaccinate against, or that they may have had problems with in the past in different, if it's layer farms or broiler farms. So it's things that because the funding for this entire project is from the research and impact development and impact fund. Yeah. So there must have been some sort of impact on the economics of the situation in Trinidad. So we focused a bit of it on the poultry side of things. And so these are just some of the viruses that can affect seabirds. Avian pox, we have different flaviviruses that are tick-borne. A lot of the birds we handled had mites and they had ticks and so on. Newcastle disease, and then of course Avian Influenza. So you'll probably know this already, and Avian Influenza is taking over the news many places. It's an Orthomyxovirus with subtypes based on the Hemagglutinin and Neuraminidase surface glycoproteins, those same proteins that stick out on the surface of the virus. Right, and then of course there's a lot of genetic diversity in influenza because of constant reassortments and mutations that take place. The clinical signs can vary from very mild to very severe, and a lot of the times in seabirds and in waterfowl you won't see clinical signs when there's avian influenza present. So we took samples on Little Tobago Island, even though those birds were apparently healthy, because there's still a chance that when we do test it that they do come up positive for some sort of low pathogenic avian influenza. Right, so in the situation here in Trinidad, concerning that is unknown, and we have an unvaccinated population. I just stole these two slides just to but we can skip this actually, we can skip these two. Right, so then there's the Newcastle disease virus, which is of interest as well for the seabirds and the waterfowl. It's a paramyxovirus, and it's found in both domestic breeds as well as various other bird species. Clinical signs again varies, and it depends on the host that is infected as well as the age of the bird. And so, according to what's on UWE's site in their library, last published appearance was in 1952 for Newcastle disease virus. This is just a snippet that I pulled from a newspaper article, but it's not in Trinidad. It's in 1997, I think, actually. But it was, it was a nice short article to show how they had a die-off of some cormorants, and they were wondering if it would affect the poultry in the area. So sometimes in this case, it did not affect the poultry. The strain of Newcastle disease that affected the cormorants was not virulent to the poultry, but it can be the other way around, so we never know what's going to happen, and it's important to be constantly watchful of what's occurring. And some of you might have come across this one as well, which is because of the IES threats group. There was a post about a sick tropic bird that was found in juvenile red bill, that was found, I think it's in St. Eustatius. Yeah, so you can go ahead. So that was, that's what the sick tropic bird looks like, and it appears as though that population, it's doing some damage there. I don't know what's going to become of that bird, if they euthanize it, okay. But it would be different. And then that's the next thing too, even though we're doing some work, or we want to do work in the amongst the wild bird populations and seabirds, if we do find things, in terms of treatment and whatnot, it'll be hard to administer those things. I just wanted to go through this. It's just a paper. I mean, if you all can pull it up, it'll be good to look at. But it's a paper that shows, how different environmental drivers can influence outbreaks in poultry and in wild birds, right? So they looked at some spatial patterns, and this is in Europe actually, and it identified environmental correlations in the various outbreak situations that they have. You can go ahead. But what I found interesting is, as you can see, the crosses are poultry and the red dots are wild birds. But as you can see, there's a lot of interaction. There are a lot of zones where you have wild birds and you have the poultry interacting. Most of the time it's because of there's some free-range poultry farms. There are shared water sources in some areas, be it a lake or streams or whatnot. And as you can see here, this is the same study, but it shows the risk. They've graded the risk, and you can see along the coastal areas and areas where you find seabirds, and that tends to be where interactions with the poultry in those areas. So why this research? We really want to get some baseline data going here in Trinidad. I think we more than have the capacity to do it. You know, there are lots of lecturers and there are lots of persons interested in getting this work done and getting it published. And then of course it's going to help, it's going to allow us to develop better control measures and prevention strategies, policies and whatnot concerning how people interact with certain birds. It's going to allow us to do some disease status declarations, you know, on OIE, FAO, certain organizations. And this research, of course, it's kind of bridging the gap between a couple of divisions. And my supervisor now he's just gotten himself a big grant for 100-something thousand euros for One Health. I don't know if you all saw it a little bit in the newspapers. And, you know, we're trying to do things that are going to impact and cover public health issues, vet health issues, as well as environmental and wildlife issues. So if you all have any areas of interest, you know, you can always put it to him. He's always, you know, willing to hear. So I don't want to leave you all, you know, a little fearful what we're going to find. We're not expecting to find any high-path avian influenza in Trinidad. Just because if there was some high-path avian influenza already, you would probably see signs. You'd see die-offs. You'd see major die-offs. Not total die-offs. But that was something that, you know, excited us until we found out that. Yeah. So why did people die? There was a poisoning. I think that's what they announcing. It will be interesting to see over time, you know, just like with the birds, you know, their travels and their migratory patterns and how their numbers fluctuate and whatnot. It would be also interesting to see that in the virology aspect of it. And of course, we want to be able to identify or know when viruses are coming from different areas. I mean, the USA, they do a lot of research and a lot of studies. They have lots of published data on viruses in blue-winged teal and some of their other bird species. And so it will be interesting to know what comes down here and what interactions the migratory birds have with the local birds and, you know, what could be passing. So the main challenge, of course, with this project, the wild bird side of the project, the seabird side of the project, is capturing the birds. And, you know, I'm very grateful to Dasha and because he allowed me to get a bit of samples. We're not harming the birds in any way, although Dasha showed you all those horrific pictures. Just doing some fecal swabs, some oral swabs, and in the future we might bleed some of the birds. We took feather samples as well, which are all pretty safe. So if you all are able to, you know, share your knowledge, I'll be very grateful for that. Because there are lots of things I don't know, or, you know, in terms of areas that are accessible for sampling. And of course, I mean, it was a pleasure bringing this information to you all, and I'd love to hear if you all have any questions. And Dasha, any questions as well.